Okay, so this is the good cell. This is the non-HIV infected cell. And we looked up the composition. It said 95% HB, 1/3 of the total cell volume. 1/3, this 95% HB is 1/3 of the total cell volume. So that means there's two other parts. Now, it talked about the outer layer of the red blood cell. It talked about how it was carbohydrate and that it had what we call surface proteins. And that's a protein encasement that's protecting the cell, of course. It talked about its membrane skeleton, which is, encasing this cell. And it talked about how it was a phosphoric lipid bilayer. So I know you doctors probably don't think of blood in this way. Maybe when you do the CBC, you say, well, the phosphorus is high or whatever. But so we have the lipid, also the fatty, we have phosphoric and lipid, right? And this comprises the bilayer, right? And it is also 12%. It's 12% cholesterol base, which is interesting, right? So we're talking about the composition of a normal, healthy red blood cell. Okay? So if we were just hypothetically saying making a cake, we would have to know the ingredients in order to take out bad things or place good things in. Medication either has to remove the bad things or it has to put in good things. Vitamins put in good things. Antibiotics and things like that take bad things out. So we're trying to figure out what specifically does the HIV RNA do to this cell. So we went to look what HIV parasitic intruders do. That's just my reference to them. HIV RNA parasitic intruders. They obviously ride on the RNA of the good cell. So RNA is a part of the body. So the body will not reject normal RNA strands, right? If the function of the body is to join itself to other good RNA or just other RNA, it's not going to reject the RNA housing of HIV parasitic intruders. So, because the body will not reject RNA, we have to say what is riding on the HIV parasitic intruders, that RNA. So we learned that there are virusoids, ribonucleic strands. Now the body accepts ribonucleic strands. But it said something about these ribonucleic strands that ride on the RNA, on the healthy RNA of a cell, which are HIV parasitic intruders, and it said that they are encased in protein structures. So here now we have our viral parasitic intruders riding on the good RNA, but they are also encased in protein structures. So we have virusoids, viroids, and ribonucleic strands encased in protein structures, right? Now, we know we have several different types of strands of HIV. So these protein structures could be varying, or the virusoids could be varying. The ribonucleic strands could be just mutated and protected by protein encasements. And the viroids, definitely are some sort of protein or enzymatic mutation. That's what these things are. But guess what? These things exist outside of the body. Now, we determined when they ride on the blood, whether the blood is infected or not. When they ride on infected blood, they have a life limit. They have like 4 seconds or something. where they have to intrude the protein encasement of the cell in order to attach themselves to the good RNA. Okay? So that's when they exist solo to their host. Okay? Now, these things, which are the HIV parasitic intruders that ride on the good RNA and introduce themselves through bad infected RNA, are said to be mutative. So they have great replication ability. They're said to be viral, and more than what kind of virus, it's the nature. It's not bacterial, it's not clinical, it's not germicidal, it's not fungal, it's viral. And viral is one of the biggest challenges to healthcare because viral is creative. Like, it is unhindered chemistry when it's set into motion, and you really have to know what the virus is and what it's riding on to stay alive in the good tissue or in the body. Okay, so then we also know something that is kind of disturbing to me anyway. It's protected by protein encasements. So this HIV parasitic intruder is actually using good science to invade the body. It is encased by protein structures, protein peptides or whatever, protein structures, and it's highly replicable again. It can mutate. Mutate means it can change and morph, and it can also duplicate itself, okay? And the reason it has this power is because of the protein. The protein and the ribonucleic strands. And all of these things are actually part of chemistry, and they ride on the good RNA. So if we were going to try to heal the body of HIV exposure, I'm sorry, I'm in a dorm, it's dark, and I'm trying to use my phone as a light so you can see. But medicine then would have to do a few things. Medicine would have to define what these virusoids are, what these ribonucleic strands are, what the the protein peptide structures are encasing them, what the viroids are, and the proteins that these ride on or that protect them so that they can not only invade the good tissue, but that they can encapsulate themselves either within or attach themselves or protect themselves as they grow within this good tissue. Okay? So, right now, the doctor said that medicine is actually stopping one facet, which is the mutation. So, it's stopping it from spelling itself different. If it was generally ABCDE, it's stopping itself from renaming itself to ABCJLK, right? Or ABCNYP, right? So here, and it's also stopping itself from changing its name, it's stopping itself from being able to replicate. So then the symptoms that it would cause by being mutative, not understood, in this good tissue or replicable, the symptoms subside because the symptoms are caused by foreign instruction in the cell. The cell is designed to work a certain way. It has a certain God-given instruction. It has a certain protection about it. And this is introducing foreign, normal cellular matter that has been infected, mutated, filled with germs, virus, fungus, or what have you, whatever it's determined to be, and it has invaded the normal tissue. So now, what theory then could be happening with the onset of the HIV exposure? of course, penetration of the cellular protein encasement. And this protein encasement, my thoughts were on that were that if a cell's normal healthy function required, let's just talk about the encasing, just hypothetical, this is ignorant but hypothetical, 5 different proteins encasing the outer core being made up of 5 different proteins, right? And so this then introduces maybe 7 more, and the proteins they join to each other, and now this has been changed. So if, let's say, medication were to come into the cell, and now there's like 12, this has hardened with a encasement of 12 proteins because of the virusoids or the viroids or the mutated ribonucleic strands, then the medicine is ineffective. This would be something that we would consider to be genetically altered and resistant to medication, right? Or say within the cell, a certain function, like they said about the nucleus, having 2 points where malaria kills the splicing function of these points in the cell, the ANI1 and the PNN, right? So it kills the splicing of the RNA matter in the nucleus so that it can't maybe increase white cell production. So that's how malaria attacks, right? So let's just say that these things rode on those portions of the nucleus. If it had already been demolished by malaria, and it was an additive function, then the cell would not pick up HIV because whatever was in the ANI1 or the PNN was diminished. So not only will this part of the nucleus not provide splicing to increase immune function, but it will be impaired. So if this added to infect, it would be in a deficit to infect because this nucleus is impaired. So penetration of the cellular protein encasement on the core or within the good tissue is one of the things for sure that is going on. And it is the change of the protein content, volume, and probably composition, chemistry, that is causing the planting or the attachment, because the protein's going to stick together, okay? Then the changing or the adding to the protein encasement. Like we said, protein could be taking the numbers up or taking the numbers down. It could be a different type of protein that cancels another protein in the good cell. And then addition of the RNA strands. Now, RNA, ribonucleic matter, is a part of the human body. So the body is not going to reject RNA. It's not like the body will reject blood. But what happens is if the person gets the wrong blood, they get a rash, they get sick, right? The body is not rejecting blood itself from another source. It's rejecting the type of blood, right? So that is the changing of, the RNA structure, content, volume, or what have you, and that's bringing on sickness or is bringing on symptoms, okay? Then addition of RNA strands in the healthy cells. So the cell has already been defined by divinity. A healthy red blood cell has thus and so, it operates in a certain way. If you add a ribonucleic strand that is viral in nature or parasitic in nature, now the number of RNA strands in the cell has changed, and that brings on sickness. Fast-growing protein viral strands, when the protein multiplication or chemistry becomes viral or is virally motivated. This girl always competes with me. Please excuse me, I'm in a dorm. Chemistry respellings, that's what I call it. That's not scientific, but that's what I call it. It's like when you have one carbon, two oxygen, and one nitrogen, instead of it being CO2, now it becomes CO2N, right? And that's a difference in spelling, right? So we also noticed that it had phosphorus tissue and lipid within this cell, right? So these proteins then are affecting the phosphorus structure or the phosphorus composition and the lipid composition. So whenever you alter the DNA of the good RNA structure or the good cell, and excuse me if I'm using the wrong words, I'm not a doctor and not a scientist, sickness happens, right? So we have to understand medication then, the aim of medication then is to not only determine what these are, maybe based on strand or strain, but to stop these things from having power within the tissue. And like she said, they are stopping mutation, they're stopping replication, but they need to stop the math. And the math is because maybe some of these viruses or parasitic issues are an increase in ribonucleic strands or increase in protein in the cell. The cell is not accustomed, not just being viral, not just being bacterial, not just being fungal in a place where it should not be, but perhaps being too much or being canceling out the good numbers. And so that should be the aim of medicine. Number one, to determine what these things are, right? So, yeah.